Usually when we talk about getting a medical diagnosis, there is a very comforting expectation of precision.
Oh, absolutely. You want a clear answer.
Right. Like you break your arm, the doctor puts you in an X-ray machine, and boom, there it is.
Yeah, the jagged white line on a glowing screen.
Exactly. The problem is visible. But when you step into the world of mental health, that really satisfying visible proof just completely vanishes.
It does. It becomes incredibly murky.
Yeah, suddenly the diagnostic landscape is based entirely on, self-reported feelings and behavioral observations. We've just sort of accepted that we can't put anxiety under a microscope.
You've had to, yeah.
But there's this striking May 2026 article from Science Daily, and it details a breakthrough from UC Davis Health that was published in Molecular Psychiatry. And it suggests that, well, an X-ray equivalent might actually exist.
It is a massive paradigm shift. I mean, for decades, the underlying chemistry of anxiety disorders across different diagnoses has been treated as this sort of black box. We knew the emotional outputs, obviously, but the physical structural footprint inside the brain was just, well, elusive.
And finding that footprint is so critical because the stakes here couldn't be higher. We take a stack of complex research on every deep dive to extract those vital aha moments. The goal is so you can be the most well-informed person in the room without the information overload. And this particular deep dive feels urgent.
What is urgent?
Yeah, because anxiety disorders are the most common mental illness in the U.S. They affect about 30% of adults.
Which is staggering when you think about it.
It's nearly 1 in 3 people you interact with daily. Yet, according to this research, a huge portion of them simply aren't receiving adequate treatment.
Well, and the current treatments often involve a lot of trial and error.
Yeah, finding the right medication is tough.
Exactly. Largely because of that missing physical footprint.
Yeah.
But the core breakthrough from this UC Davis meta-analysis, it gives us a completely new target.
A real physical target.
Yes. Scientists have finally identified the first clear, consistent chemical pattern in the living brain that links across multiple different anxiety diagnoses.
Which is just wild.
Right. And the most surprising part is that this shared chemical footprint points to a hidden deficit of a specific nutrient, like a material most Americans are actively deficient in.
Okay, let's unpack this.
Yeah.
Because calling it a nutrient deficit makes it sound like, I don't know, we were just talking about someone forgetting to take their morning multivitamin.
Yeah, it sounds trivial.
But looking at the source material, we're actually talking about the fundamental physical architecture of the brain itself. The missing ingredient is a nutrient called choline.
Right. The researchers found that individuals diagnosed with anxiety disorders have about an 8% lower level of choline in specific regions of their brains compared to people without anxiety.
8%.
Yeah. And to understand why that matters, we have to look at what choline actually does. It's an essential nutrient with a highly diverse job description.
It does a lot. All right.
It does. It regulates mood, supports memory, aids in muscle control. But at the microscopic level, choline is a structural imperative.
Meaning it builds things.
Exactly. It is required to produce phospholipids, which are the primary building blocks of cell membranes.
So it's forming the actual outer boundaries of our cells, like it's the physical barrier.
Yes, and it doesn't stop at structure. Choline is also the direct precursor to acetylcholine, which is a really critical neurotransmitter.
Okay, so it's doing double duty.
It is. So it's simultaneously building the walls of the cell and manufacturing the chemical messengers that allow those cells to talk to each other across the nervous system.
That is fascinating.
Yeah.
But the researchers didn't just find like a generalized drop in choline across the entire brain, though. They pinpointed a very specific location.
They did.
They saw this 8% deficit was most pronounced in the prefrontal cortex, which is basically the brain's executive control center right behind the forehead.
The CEO of the brain, basically.
Yeah, right. It's the region responsible for our high-level emotional control, our complex decision-making, planning, and just, regulating our outward behavior.
It is the region that essentially looks at a chaotic situation and decides how to rationally respond.
Like separating a real threat from a fake one.
Exactly. If your emotional centers are sounding an alarm, the prefrontal cortex is the manager stepping in to assess whether the threat is real and how to handle it.
So if we think about the prefrontal cortex as a major corporate office trying to manage a constant stream of crises, choline isn't just the coffee keeping the office workers awake.
No, not at all.
It is the actual drywall making up the physical offices.
Hmm.
And it's the raw copper in the communication cables holding the entire building's network together. Does that track?
What's fascinating here is that that analogy is spot on. And taking that corporate office analogy further, imagine if that office was incapable of manufacturing its own drywall or its own networking cables.
Like they couldn't just produce it on site.
Right. It had to import every single piece from an outside supplier. You see, the human body can synthesize a microscopic amount of choline in the liver, but it is nowhere near enough to meet the biological demand.
So we have to eat it.
The vast majority of it must be imported through our diet. Our brains absolutely rely on the food we consume to maintain those structural walls and communication lines.
Okay, but if this structural material has to be imported and the deficit is happening deep inside the prefrontal cortex, it raises a major methodological question for me.
How they measured it.
Yeah. How did these researchers figure out what's happening inside a living, functioning brain? I mean, you can't exactly biopsy the prefrontal cortex of 300 living patients to measure their lipid levels.
Definitely not.
They must be using some kind of advanced neuroimaging to get a chemical readout.
The methodology is the backbone of why this study is so groundbreaking. The lead researchers, Richard Maddock and Jason Smuckany, they utilized a technology called 1H-MRS.
Okay, what does that stand for?
It stands for proton magnetic resonance spectroscopy.
Spectroscopy. So we aren't just looking at structural shapes like we would with a standard MRI. We're looking for actual chemical signatures.
Precisely. Standard MRIs are fantastic for giving us a high-resolution map of anatomy. Like, if there is a tumor or a structural lesion, an MRI will show it.
Right, the physical shapes.
But 1H-MRS takes advantage of the fact that protons in different molecules spin and behave differently when subjected to magnetic fields.
Oh, that's clever.
Yeah. By tuning into those specific resonant frequencies, this technology can safely and non-invasively measure the exact concentrations of neurometabolites.
Meaning the chemicals involved in brain metabolism.
Exactly, right there within the living tissue.
So it's essentially taking a real-time chemical inventory without ever breaking the skin.
That's a great way to put it.
And what gives this finding so much weight is the sheer scale of the data. I mean, this wasn't a single trial with 20 undergrads in a lab.
No, not at all.
The UC Davis team conducted a massive meta-analysis. They pooled and reviewed data from 25 previous studies that had utilized this specific 1H-MRS technology.
It was a huge undertaking.
Yeah, they ended up analyzing the brain chemistry of 370 people diagnosed with anxiety disorders and compared it against a control group of 342 people without anxiety.
And pooling that data is what allowed them to see through the noise. In individual, smaller studies, subtle chemical differences can easily be dismissed as, statistical anomalies.
There's a fluke in the data.
Right. But with over 700 participants, the pattern became undeniable.
I do want to push back on the math for a second, though.
Sure.
Because we need to put that 8% figure into perspective. To a layperson, an 8% reduction sounds like a rounding error.
It does sound small.
Right. Like if my phone battery drops from 100% to 92%, my user experience doesn't change at all. I don't even notice it. Is an 8% drop in a single nutrient really enough to fundamentally alter how a human being processes profound stress and emotion?
It's a totally fair question. Richard Maddock, the senior author of the study and a psychiatrist who has spent decades analyzing how anxiety affects brain function, addressed that exact skepticism.
What did he say?
His direct quote from the source is, an 8% lower amount doesn't sound like that much, but in the brain, it's significant. We have to remember that the brain does not operate like a phone battery.
It's not just a fuel gauge.
Right. It operates as a highly calibrated, interconnected ecosystem.
So it's less about a fuel tank being slightly less full and more about really delicate chemical equilibrium being thrown off balance.
Exactly. The brain relies on incredibly fine margins of homeostasis. A chronic 8% deficit in the structural and signaling materials of the prefrontal cortex means that millions of neurons are operating at a suboptimal level.
They're struggling to keep up.
Yeah, the transmission of those acetylcholine messengers becomes less efficient. And what makes this 8% drop so powerful as a finding isn't just the percentage itself, but its relentless consistency.
The meta-analysis did look at other chemicals, right? It wasn't exclusively focused on choline.
Oh, they examined a wide range of neurometabolites. For instance, they noted reduced levels of cortical NAA, which is a marker related to general neuronal health and function.
Okay.
The drop in choline-containing compounds was the single clearest, most consistent signal across the board. And crucially, it was a transdiagnostic finding.
Meaning it wasn't isolated to just one flavor of anxiety.
Correct.
They saw the same 8% deficit in generalized anxiety disorder, panic disorder, social anxiety disorders, and specific phobias. It is a shared biological denominator across all these distinct clinical diagnoses.
Which is revolutionary for psychiatry. We are moving away from categorizing these disorders solely by their behavioral symptoms and starting to categorize them by their underlying neurochemistry.
But if this 8% drop is mathematically significant and, chemically destabilizing, we had to ask the causal question. Why is the choline disappearing in the first place?
That's the big mystery.
Right, like if people with anxiety are eating the same general diets as the control group, why is their prefrontal cortex running such a massive deficit?
To answer that, we have to look at the mechanics of fear processing.
Yeah.
We established that the prefrontal cortex is the logical manager.
The CEO.
Right. But it is in a constant dynamic dialogue with another brain region, the amygdala.
Ah, the amygdala.
The amygdala is our primal alarm system. It is constantly scanning the environment for threats and shaping our base sense of safety.
So in a healthy system, the amygdala detects a loud noise and says, threat. And the prefrontal cortex evaluates the context and says, stand down, it's just a car backfiring.
That is the ideal threat appraisal process.
Wow.
But in anxiety disorders, that system is chronically dysregulated.
It's out of whack.
Exactly. The amygdala becomes hyper-reactive. Every day concerns an email from a boss, a crowded room, an upcoming flight. They trigger a profound survival response. The alarm bell is stuck in the on position.
And from a chemical standpoint, that alarm bell is ringing via neurotransmitters like norepinephrine, which drives the body's fight-or-flight response.
Yes.
So heart rate spikes, muscles tense, attention narrows.
And people with anxiety disorders often have chronically elevated levels of norepinephrine. They're essentially living in a perpetual state of heightened physiological arousal.
Exhausting.
Truly. And this is the crux of the researchers' hypothesis for the missing choline. The UC Davis team suggests that this chronic, hyper-aroused state drastically increases the prefrontal cortex's demand for choline.
Here's where it gets really interesting. Because if the amygdala is flooding the brain with false alarms, the prefrontal cortex is being forced to work overtime.
It's working frantically.
It is desperately trying to synthesize enough acetylcholine to send inhibitory signals to calm the system down. Mechanically, it is like driving a car with your foot slammed on the accelerator while simultaneously ripping the emergency brake up as hard as you can.
That's a perfect image.
The engine is just roaring, burning through its fuel at an accelerated rate to manage the competing signals.
And the metabolic demand on the tissue is immense. Under that kind of chronic stress, the prefrontal cortex is burning through its available choline faster than it can be replenished.
Wow.
And if the dietary intake of choline isn't high enough to meet that sudden massive spike in demand, the brain might actually start cannibalizing its own cell membranes to get it.
Wait, cannibalizing?
Yeah, it starts breaking down the phosphatidylcholine in the cell walls just to keep the nerve signaling going. Hence, the measurable 8% deficit inside the tissue.
That is wild. The brain is literally tearing down its own drywall to keep the communication cables firing.
Exactly. It's sacrificing structure for function.
So if the tank is running low because of this intense chronic fight-or-flight drain, the immediate practical question for anyone listening is how to intervene.
Right. How to fix it.
How do we refill the tank? Since our bodies cannot manufacture a meaningful amount of choline endogenously, we have to look at our diet.
And the study highlights several specific foods that are naturally rich sources of dietary choline. The highest concentrations are typically found in animal products.
The article specifically points to beef liver as one of the most potent sources, followed by egg yolks.
Yes, eggs are a huge one.
It also lists beef, chicken, fish, soybeans, and milk. And it mentions that certain omega-3 fatty acids, particularly the types found in salmon, are highly effective at supporting the brain's choline utilization.
But there is an alarming context surrounding this dietary list, and it's a warning straight from Dr. Maddock.
Oh.
Based on broad nutritional surveys, the vast majority of people in the United States, adults and children alike, do not consume the recommended daily amount of choline. Yes. We are looking at a population-wide dietary deficit of the exact same structural building block that our brains desperately need to regulate stress.
So what does this all mean for you as a listener? If you are dealing with chronic anxiety or even just feeling incredibly stressed about an upcoming project, should you immediately go out, buy a massive bottle of choline supplements, and just eat a carton of egg yolks expecting to cure your anxiety.
The researchers are incredibly firm on this no.
Okay.
You should not self-medicate with high-dose choline supplements. It is really tempting to look at a chemical deficit and assume that simply flooding the body with the missing ingredient will fix the problem, but neurobiology is rarely that linear.
But why wouldn't it be? I mean, if the brain is tearing down its drywall because it lacks the raw material, shouldn't giving it a surplus of raw material solve the supply chain issue?
Well, because of the blood-brain barrier, for one.
Oh, right.
Consuming a nutrient doesn't guarantee that it will efficiently cross into the central nervous system and be taken up by the prefrontal cortex in the exact way we want.
It's not a direct pipeline.
Exactly. Furthermore, introducing massive amounts of a single precursor can sometimes throw other delicate metabolic pathways out of balance. But more importantly, we have to look at the nuance in the subsequent research.
Right. The article mentions that since this initial imaging study was published, other researchers have dug into the actual dietary data to see how choline intake correlates with mental health out in the real world.
They did. They looked at large-scale nutritional data to see if people who naturally ate more choline had better mental health outcomes. The findings were fascinating, but, well, mixed.
Mixed how?
They found that in adults, higher dietary choline intake was consistently linked to lower odds of experiencing depression.
Which is a huge finding on its own.
Absolutely.
Yeah.
But in those same dietary studies, they did not find a statistically significant association between higher choline intake and lower levels of anxiety.
Wait, let me make sure I have this straight. The brain imaging proves that people with anxiety have a physical choline deficit in their prefrontal cortex. But the dietary data suggests that simply eating more choline doesn't necessarily protect you from anxiety, even though it might protect you from depression.
That is the current state of the data, and it highlights a really critical scientific distinction. If we connect this to the bigger picture, the UC Davis imaging study provides a phenomenal biomarker.
The 1-HMRS scans.
Right. They prove that a measurable physical deficit exists inside the brain. It is documented and real. However, the imaging study does not prove causality.
So it doesn't prove that a poor diet caused the anxiety in the first place.
Precisely. It is entirely possible that the choline deficit is a symptom of the anxiety, a result of the brain burning through its resources to handle the chronic stress, just like your car analogy. And simply adding more gas to a car that's constantly running at the red line with the emergency brake on, while it might not fix the underlying mechanical issue causing the engine to rev.
That makes total sense. To truly know if dietary interventions work for anxiety, scientists are going to have to run controlled, double-blind clinical trials. They need to intentionally alter choline intake in patients with diagnosed anxiety disorders and use these 1-HMRS scans to measure if the brain chemistry actually normalizes over time and if symptom relief follows.
And until those clinical trials are completed, nutrition remains just one piece of a much larger therapeutic puzzle. Ensuring you get adequate choline is fantastic for your overall brain health, your cell membranes, and potentially your risk for depression.
Oh, sure.
But the source is adamant that dietary changes are not a substitute for professional mental health care. Therapy, behavioral interventions, and proper medical guidance remain the frontline treatments for anxiety disorders.
So we have discovered an incredibly important piece of the map, but we haven't charted the entire territory yet.
Exactly.
To synthesize all of this for you for generations, there has been an unspoken stigma surrounding mental health. The idea that anxiety is just an emotional weakness, something purely in your head that you should just be able to think your way out of.
What is so damaging?
It really is.
Yeah.
But what this deep dive reveals is that anxiety has a physical, heavy chemical footprint.
The discovery of a consistent 8% choline deficit in the prefrontal cortex across multiple different anxiety disorders completely changes the landscape.
It really validates the experience.
It moves anxiety out of the realm of abstract emotion and grounds it in measurable physical neurochemistry. It proves that the brain is structurally fighting a battle.
And even if a choline pill isn't going to magically rewire your amygdala overnight, the core realization here is incredibly empowering.
I completely agree.
Our brains are not ethereal computers. They're biological organs. They require literal physical materials to construct the communication cables that manage our moods. Evaluating your baseline nutrition isn't just about waistlines or heart health.
No, it's about providing the architectural supplies your brain needs to handle the friction of daily life.
It forces us to respect the profound physical connection between what we consume and how resilient our nervous system can be.
It really does.
We started this deep dive by comparing the murky world of mental health to a clean, definitive X-ray of a broken arm. Thanks to one HMRS technology and researchers looking past the symptoms into the chemistry, we finally had the beginnings of that X-ray for anxiety. And as you go about the rest of your day, I want to leave you with one final thought to mull over. We now have concrete proof that our brains require specific physical molecules, like choline, just to mathematically process abstract feelings like uncertainty and fear. It makes you wonder how many of our other fleeting daily emotional states are sudden bursts of patience, our spontaneous joy, or our lingering unexplained frustrations are secretly being dictated by the chemical inventory of whatever happens to be sitting on our grocery lists.